And I welcome you to the third program of the 2024 Islesboro Forum Speaker Series. I'm Gail Jackson and the moderator of today's program. As is customary, we will hear from our speaker for about 25 to 30 minutes and then have time for Q&A afterwards. And he has graciously said that he likes the question and answer part the best, so he'll be willing to extend that if it seems to be the thing to do. We are proud to be in our 32nd summer since we started the forum, and we're going strong. We will record tonight's program and post it in several days at islesboroforum.org. So spread the word to friends who were unable to make this live presentation. We already have the two preceding programs posted at islesboroforum.org, so you can find them there, and we'll have this one up shortly in a few days. I always take a moment to ask people to consider making a contribution. We do have expenses for our very capable technical producer, among other things, some trips on the Quicksilver to bring speakers back and forth, publicity, etc. We suggest a modest $10 per program. There's a basket right there. You can make a check or put some cash in. There are instructions on the website for where to send the check. Turning to our program, we are excited to present Craig Venter, a relatively new island resident, also founder, CEO, and chairman of the J. Craig Venter Institute. A biologist, he's renowned for his contributions in genomics, including sequencing the first draft human genome, the first complete diploid human genome, and construction of the first synthetic bacterial cell. His current research focuses on the human genome and how it relates to disease and health. In addition, he has devoted countless hours and sailing of the high seas to understand genetic diversity in the world's oceans. That gripping tale is set forth in his book, The Voyage of the Sorcerer II, the expedition that unlocked the secrets of the ocean's microbiome. Courtesy of Main Point Bookstore here on the island, that book is available here today and will continue to be at the bookstore in the weeks ahead. He has written several other books, including his memoir. Miriam has the books on the table in the back if you want to avail yourself, and Craig would be happy to autograph them for you. I'll just finish the introduction of Craig by pointing out two highly significant honors that he has received. He's a recipient of the 2008 National Medal of Science and a member of the National Academy of Sciences. Venter is quite simply a star in the world of science, and you will now be privileged to hear from him directly. Please welcome him for us.
Well, Gail, thank you very much for such a nice introduction, and this increases by about 20-fold the number of people I've seen on Islesboro. So it's we're out on the tip of Turtle Head, so we don't get a lot of visitors out there, which is part of the attraction, but that's it's great. So I'm delighted to talk to you about my latest book, The Voyage of Sorcerer II, but it's about sailing and science, and I often get asked what came first, sailing or science. And sailing definitely came first. In fact, I have an odd tale of how I started sailing. I think I had just turned 20. It's so long ago I had trouble remembering. and I was in Vietnam, and I did a surgical procedure on the harbor master of Da Nang, who went on R&R to Bangkok and came back with the letters spelling out Mary on his fingers. And he was going on a second R&R to see his wife in Hawaii, but her name was not Mary. So he was very anxious to have surgical removal of his tattoos, but being a war zone, he was turned down for, they don't do elective surgery, but I felt sorry for him, so I removed his tattoos and did skin grafts. And he convinced his wife he was a war hero instead of a victim of the houses in Bangkok. But he was so grateful, he gave me a 19-foot Lightning sailboat. And he asked if I knew how to sail, and I said, well, of course I do, but because I didn't want him to not give it to me. So I actually learned to sail by taking it out through the surf on what's now known as China Beach, and sailing around there. So I contend I had the only Lightning with bullet holes in the sail. So that started my sailing career, but one of the things that I thought of and read a lot about, reading about sailing, was sailing around the world. And a lot of people think about it, talk about it, very few people actually do it. And I had the opportunity with a science question I wanted to ask to combine the two things. And some of my colleagues think the most brilliant contribution and idea I had was combining sailing my own boat around the world while getting paid and doing science. So it was a great construct. But the science started after Vietnam, but I was a researcher at the National Institutes of Health, and I invented a method for very rapidly discovering human genes. And a lot of the work, because it was using new techniques for sequencing DNA automatically with a machine, it generated a lot of data, so computational science became a very key part of what we were doing. And we developed a new algorithm for assembling DNA sequences. In 1992, when I started my institute, I was attracted out of NIH. I was given a $70 million commitment over 10 years to start my own research institute in exchange for the IP going to a new biotech company called Human Genome Sciences. But I wanted to apply this new technique. No genomes had been sequenced yet. And the way this technique works, imagine taking the Sunday New York Times, like 20 copies of it, putting it through a paper shredder, and then taking what's in the paper shredder and reassembling one bona fide copy. And so that's what our algorithm does, but DNA sequence is much simpler in many respects because it's just a 4-letter code, A, C, G, and T. And each of you have 6 billion letters in your genome, so there's a lot of them. But the strings are very unique. And so just by finding overlaps of 16 to 20 letters, the computer could re-piece together all these fragments. The machines at the time produced stretches of DNA 500 letters long. So imagine trying to reassemble 6 billion letters from pieces only 500 letters long. So that's what the algorithm did nicely. And my team in 1995 ended up sequencing the first genome in history of a bacteria called Haemophilus influenzae, and it sort of shocked the world because nobody believed that the computer approach would work for this, that DNA was too complicated and it needed tens of thousands of scientists to put these things together. I didn't actually know how good the algorithm was until we were sent a clinical sample from Norway of a patient that had a bacterial infection, and we sequenced it, and the algorithm actually assembled two completely separate but related species. So the mathematics are so precise, and each of your genomes and the genome of every species on the planet is mathematically unique. So we're able to reconstruct things even from complex mixtures. And that's where I first got the idea. I was doing a lot of sailing at the time. I was sailing between Washington, D.C. and Cape Cod, and then frequently down to the Caribbean, usually via Bermuda. And there was a paper published in the Proceedings of the National Academy of Science saying that the ocean was pretty much, very low diversity of life. The Sargasso Sea was viewed as this ocean desert. It's actually a bad metaphor. We have a ranch in the California desert east of San Diego, and it's highly diverse with life, but people assume that it's not. My idea was simply to take 200 liters of seawater and filter out all the viruses and microorganisms. So there's different filters you can buy that have very small pores, down to 0.22 microns. In fact, some people sterilize water that way just by filtering it and filtering out all the bacteria. But we did the opposite. We took the filters and then we isolated the DNA off the filters and then just sequenced everything that was there. So it was a random mixture. But the scientific community was sure we would find very little. Microorganisms in the Sargasso Sea were numbered one at a time as they were discovered, and they were up to 11. So it was SAR 11 for Sargasso Sea 11. The whole history of microbiology before our approach in genomics was if you couldn't culture it, it basically didn't exist. So all the species that we know that cause infections, all the species that were understood to be part of the world were because people could take a sample of soil or seawater and get it to grow in the laboratory. It turns out they were missing 99% plus of biodiversity by having that limitation. But, other than the microscope, those were the only tools that existed to understand the microbial world. Most microbes were discovered because they caused infections in people, and so there was a reason to look for them. But it turns out we live on a microbial planet. The microbes were the first species here, and they'll be here after we're long gone. But just from one barrel of seawater, we discovered several thousand new species. And we stopped sequencing after I think we discovered 1.5 million new genes just from the single sample. Not very often does one simple idea, scientific study, change the view of people's thinking, but this did because it showed in one instance how much life was totally missed up until that point in history. So Darwin on his expedition based everything on the visual world, on his visual acuity. You know, what he saw, he characterized extremely well and came up with really an interesting theory, but he missed about 99% of the life forms that were in the Galapagos while he was there. DNA was unknown, the mechanisms of genetics were unknown, but makes it even more astounding how he was able to put together the diversity and also the uniqueness of life when it's isolated, like on an island, or set of islands like the Galapagos for a period of time. And that's how diversity gets fixed, both with human populations and with every species imaginable. So we could have just kept sampling in the Sargasso Sea and sailing to Bermuda, and I just asked another simple question, you know, what if we sample 100 miles away? Is it just going to be the same set of microbes, or will they be different? And the first sailing expedition was actually not Darwin. It was actually an expedition called the Challenger Expedition. Darwin was a naturalist. Quite often survey boats would take a naturalist, and he was just a naturalist on one of the ocean survey boats. But in late 1870s, the Challenger Expedition was the first true scientific expedition, and the first transatlantic cables were being laid, and nobody had any idea what the bottom of the ocean was. And for some reason, the brilliant scientists of the time came up with this notion that there couldn't be any life below a certain depth in the ocean. I contend most things that are considered major discoveries is because people had really stupid ideas before them of what life was. And all people like me do is, uncover what the facts of life are versus the idea. And the Challenger Expedition sent a dredge down every 200 miles around the globe, starting in Halifax, and every place, at every depth, and they discovered the Mariana Trench. They found life, and unique life. And so that was the first thing that really changed the view of diversity of life, and that it could exist at every depth in the ocean and every environment. We decided to sort of follow their route, starting in Halifax, and they went largely around the equator. And those of you who are sailors know why you would sample every 200 miles. That's roughly what a good sailing vessel can do in a day. And that's, Sorcerer sometimes would do more, sometimes less, but we decided to stop every 200 miles and collect 200 to 400 liters of seawater. And we probably have the only sailboat with a minus 80-degree freezer on board. And we would just simply put these thin filter papers in the freezer till we got into port, and then we'd ship them back FedEx to the lab where all the DNA was sequenced. And the first amazing finding was every 200 miles, 80% of the sequences were totally unique. So instead of the ocean being a giant homogeneous soup, or up here a lobster soup, it's actually millions and millions of microenvironments changing constantly at every depth. And then we have microbes that live up to 120 degrees centigrade. They live off of hydrogen, they live off of sulfur. We have ones that live in pH 11 water, so if you put your finger in it, it would dissolve your skin. People in the DOE have discovered ones in acid mine runoffs that live at pH 1, which is a fairly strong acid. Also, if you put your finger in it, it would dissolve the skin. So microbes live in every environment, and 65,000 miles around the sea, taking samples every 200 miles, we discovered more new species and species in our expedition than the rest of the history of science put together had discovered. And we couldn't name all of them because there were way too many. We thought about selling, like they do stars, but people could name their own microbe, but there were just way too many. And Gordon Moore gave us substantial funding through his foundation for the expedition, and he paid for a huge computer database to house all this data, where people have been using it around the world ever since the first collections. Now, the science part was great, and you think it'd be very easy just to sail around the world and collect 200 liters of seawater, but it ended up being a little bit more complicated. There's a thing that you may have heard of recently with some of the latest politics called the law of the sea, where countries own all the now genetic species out to 200 miles offshore. These were originally set up for fishing rights, but ever since microbes were proven and the genes in them were proven to have value, they extended it to all DNA and RNA species. So we could not just sail 200 miles off of islands in French Polynesia without having to have permits from the French government. And it turned out much more complicated. So these laws are very strange. So we sailed from the Galapagos to the Marquesas. It's the longest open ocean passage that you can do. And there's roughly a 1-knot current that moves across the Pacific towards French Polynesia. All the species, microbes, fish, mammals, anything that's in the ocean belongs to nobody and everybody until it crosses 200 miles off of the Marquesas, when all of a sudden it becomes French genetic heritage. We were just watching the surfing in Tahiti, and it's really fantastic. But it proves that France still considers French Polynesia part of France, even though French Polynesia has been disputing this for some time. So we had permits from the Tahitian government in French Polynesia to take samples. But when we sailed into Eva Ova, which is the first island when you reach the Marquesas, we were placed under house arrest by the French police, and the French Navy said that if we left the harbor that we would, well, they implied that we'd be sunk. They said they could not guarantee our safety, but you might remember not that long ago the French sunk the Rainbow Warrior in New Zealand harbor simply for protesting the nuclear blast in French Polynesia. So we had to get the State Department involved, the U.S. Navy. We had, all this over taking water samples to discover new species. Fortunately, the State Department was a huge help, and the U.S. Navy told the French that we were a U.S. research vessel and we were under U.S. Navy protection. So what they insisted that I fly to Tahiti to negotiate a new agreement with the French Polynesia government. So we had to have sign off because, well, Paris thinks they're in control. Tahiti didn't think they were in control, and we simply got caught in the crossfire. It's one of the most beautiful spots. I mean, some of the diving, we got to scuba dive in the best spots in the world, aside from doing science and meeting people. It wasn't the only time we were arrested or confronted by the police. We spent about 6 months in Australia, mostly in Brisbane. And one of our crew members was dating a local woman who was engaged before, and I had just flown back from the States. I did not stay on the boat all the time in the different countries. I mostly did the ocean passages. And I was just sitting on the deck having a beer, and a SWAT team pulled up, and a large number of armed police came and said they wanted to board Sorcerer because they were told that we had a meth lab. And it turns out the jilted boyfriend is the one who called the authorities. So it was actually a woman captain that was leading the SWAT team, and I said, well, we're a U.S. research vessel. I don't have to let you board, but if you agree to take off your combat boots, and if you want to see in any compartment, you ask a crew member to open it versus what often happens with drug raids, they rip boats apart. This woman agreed to that, and it was obviously on any boat like this, you go below, it's pretty clear there's no meth lab there. So they were a little bit embarrassed, and Jeff is the crew member that did most of the sampling that was dating this local woman. And I asked if they would arrest him and take him away just for fun, but they posed for photographs with him instead. And so that one ended well. The third time we were arrested was sailing across the Indian Ocean. There's a set of islands where Diego Garcia is part of them, the Chagos Islands. And I don't know if any of you have sailed across the Indian Ocean before, but Chagos is a very famous spot that sailors stop for a break of sailing across the Indian Ocean. And it's one of the most beautiful atolls that I've seen anywhere in the world. Nobody lives on these islands because the British, with American help, removed all the natives. You've heard of Diego Garcia. We have a B-52 air base there that we lease from the British. The natives have sued in world court for years and have won every time, but the British still won't let them back in, and we're still using Diego Garcia as an air base. We just dropped anchor, and a rib pulled up with 10 heavily armed people with no flags, no insignias, and said they were going to board us. I had some weapons on board, but I figured that this was a losing situation, trying to prevent them from boarding. And once they boarded us at gunpoint, they said they were from the British military. They took our passports, and once again we were under house arrest. Only this time they left an armed guard on board. Fortunately, I had a satellite phone in my cabin, and from my adventures and politics and dealing with President Clinton and others, I knew the ambassador to the Court of St. James, the British ambassador, and I called his cell phone and explained what had happened from my cabin, and he said, "Hold on, I'll get right back to you." And I complained that they had no flags and no insignias and no identification. He obviously went right to the top of the British government, because when they came back, they had their dress flags, and they were all in dress uniforms, but they still had all their weapons with them. But they had negotiated a deal through the ambassador that I had to sign this agreement saying I had violated this one law. The law was actually a British law from the 1800s that if sailing vessels had dive equipment on board, it had to be registered with the British government. And we clearly had dive equipment on board. In fact, I had just gone diving in this beautiful atoll. One of the things, all the beaches were scattered with pieces of red coral. I mean, it was just beautiful. And apparently you're not supposed to pick up red coral anywhere, but I had this huge bucket of it sitting next to me when they boarded, and I just casually threw my jacket over the bucket. So they never saw that, and when they weren't looking, I dumped most of it over the side. But so we actually had broken a law, but not the one they claimed. And they said if I did not sign this agreement, they would keep our passports and leave a guard there and come back in a week. And they would keep doing that, giving me time to think about it. So this was all taking place in my cabin on board Sorcerer to the ambassador on live satellite speakerphone. And so I said, "Mr. Ambassador, I will sign the agreement, but I'm signing it under duress." I said, "There's armed people in my cabin telling me I have to sign it or they'll leave." And that they were fine with that. Everything was great. And they said, they gave us our passports and said, "Okay, you're free to leave." We pulled up anchor. We got about a mile outside the reef, and my satellite phone rang, and it was the ambassador. These events always bring back emotions, but nobody wants to get calls like this. But he said, "You have to stop or they'll sink you." I always take messages like that seriously. So we stopped, and they had a gunboat behind us. And I asked what the problem was, and he said the British government won't accept an agreement signed under duress. So you can see where this is going. So he said, "I want you to think very carefully before you answer this question. Were you under duress when you signed the agreement?" And the thought going through my head was, I'm really fucking under duress now. because they were threatening to impound the boat and arrest us. It wasn't clear under what basis. But I said, Mr. Ambassador, I was not under duress. And he said, that's the answer I was hoping to hear. You're now free to leave. So, science is not as simple as people think it is, and not always as glorious and fun. But also adventures on the high seas. We got to, one of the most important things I learned by sailing around the world was you see the world as it was developed, because it was developed by sea. And so sailing into Gibraltar, sailing into the Galapagos, sailing into different ports, you see the world as it was seen until aviation. So it was a phenomenal experience and education. The science changed the world, and it also showed the peril that we're putting the oceans in. There's areas now much larger than Texas that are totally oxygen-free. The biggest one's developing off the state of Oregon, so no fish can survive there, no mammals survive there, and there's now dozens of these anoxic zones developing around the world, and that's where a lot of the toxic microbes develop, because many of them are anaerobic, so they don't need oxygen. But we described one in the book of, it actually led to Alfred Hitchcock, The Birds. It's a red tide toxin that happens off of California that when sea lions eat it and birds eat it, this neurotoxin causes them to really go crazy and attack people and do things. So there's real world and real fiction based on these things. And so the other thing we encountered were ocean currents converge off of Costa Rica, a several mile wide patch of trash, just plastic debris. There was a refrigerator floating there that one of the crew members wanted to open to see if there was beer in it. But so, we saw some of the most beautiful scenes you could ever have in the oceans, and some of the most perilous of what our future could be if we don't do something about it. So I'll end on that cheery note and be happy to take your questions. I don't know if can we turn the lights up so I can.
Yes, we can.
All right. I can see to the second row, so yeah, please. Maybe stand up so everybody can hear your questions.
Has your institute at this point been asked to examine any space samples?
So the question is, have we been asked to examine any space samples? And the answer is yes, from the space station. We've sequenced all the, not all, but several of the HEPA filters off the space station. You don't want to go to the space station if you don't have to. It's basically a bacterial cesspool. And imagine every new astronaut, particularly from different parts of the world that goes up there, is taking 20 million unique microbes with them. And obviously, they don't have plumbing facilities. I got the head of NASA very upset with me one time because I said the outside of the space station was covered with feces. Turns out it was, because they would just pump all the waste out a port. And they stopped doing that, but it has a different consequence. So they pack it now into stainless steel containers and launch it back into the Earth's atmosphere. So I tell people to be very careful when you're wishing upon a shooting star, because you may get more than you bargained for. But the diversity of microbes there, we would love to do samples from Mars. So my second book is called Life at the Speed of Light. We're talking about sending sequence information as digital information. And my institute made the first synthetic cell by actually synthesizing the whole genetic code. And so if there were species on Mars, it would take a relatively short time at the speed of light to send the species back digitally, and then we could reconstruct them in the laboratory. But Elon Musk's way is to spend billions on a rocket that can bring things back, but that's far more dangerous. But hopefully he'll get a one-way ticket. So, yes, ma'am.
I'm really afraid of shocks that kind of still am. And now I'm really afraid of these deoxygenated areas as high as Texas. And I'm listening to beggars about. So, is there any, what, in your opinion, about a scientist, how can you remediate that horrible deoxygenated thing that nothing can live in?
So, all of us can only make tiny incremental differences. But the amazing thing is when you multiply the difference you can make by 6 billion, it's a really big difference. And it's stunning that it's still politically divisive to even talk about climate change in this country when the microbes even affect the weather. So with El Nino and El Niños, it's actually measuring the chlorophyll from the microorganisms in the ocean, and the density of those predict the weather outcomes. So we can all, try to reduce, the runoff from our properties into the oceans. One of the problems, as many Mainers know, is the temperature is increasing in the ocean, and the lobsters are starting to vote with their fins and move north. So these things have real economic impact as well as social. I don't know if any of you have been to the Seychelles with the beautiful white sandy beaches. Those are a result of a 1 degree centigrade temperature change in the Indian Ocean, which killed off a single microbe that was a symbiont with the coral that kept the coral alive. As a result, the coral died. It turned into all these white sandy beaches. But a temperature change of 1 degree is a big deal in the oceans. And we're facing 5 or more degrees change in the coming decades. So it's all about all the things we normally do. Even driving electric cars isn't the solution. Depends on where the electricity comes from. If it comes from a coal-powered power plant, you're driving a coal-powered vehicle. If you're charging it from solar power, it's totally a different equation. So it's a matter of just being thoughtful and thorough about every little step that we take, and contribute to the environment, and help make it so it's not a political issue before it's too late to change it. So thank you for your question. Yes, ma'am.
You've accumulated a very large database of incredible information. How are you, or how is one thinking about extracting from that database information about new forms of life or energy utilization or building materials or structural changes and all those things that are embedded in that?
Yeah, no, it's a great question. We primarily did it by making the data freely available to everybody that's interested around the world. There's literally been close to 1,000 scientific papers published by scientists using the data to make totally new discoveries. We characterized the virus environment, and there's so few viruses that have been characterized. The sequences that we discovered didn't match anything in the existing databases. They're just totally new. They just look like alien life. One of the most important discoveries we made that sort of creates a philosophical problem for me is we found the viruses, for example, with the photosynthetic microbes in the ocean that contribute about 50% of the oxygen we breathe. The viruses actually upgrade the DNA software of their host. So viruses don't want to kill their host or they lose their ability to reproduce. So in the oceans, they actually bring in new versions of the genes to make their host even more robust. So does that mean viruses, which are not self-reproducing on their own, are they at the top of the intellectual food chain? Or so it's really interesting. If viruses are trying to keep us alive, that's very different than the picture that we get with pandemics, like we all just went through. So the data is available online through government databases and other sources, and people are using it every day to make vast new discoveries. So we couldn't be more pleased with it. Yes, sir.
First of all, it's an honor to meet you. I'm a oceanography student at Maine Maritime Academy. What advice would you have for inspiring biologists?
Graduate, get your degree. Is that your dad next to you? He just gave me a thumbs up. That's certainly the best place to start. But look, the amazing thing that I think my story shows is how you can ask very simple seeming basic questions and make more discoveries than have been made before. So any student, anybody could go out here, whether it's one of the ponds, off your beaches, and take a sample of seawater, and if you had access to a DNA sequencer, you could make tens of thousands of new discoveries. Just taking a sample of soil, I mean, everything. We're in a microbial world. You have about 20 million microbes living inside of you. Most people are about 5 to 8 pounds of microbes. So there's a lot to discover, but don't be afraid that, the simple, sometimes seeming stupid questions are usually the most profound. So don't be afraid to ask them. Yes, ma'am.
So can I ask you, related to that topic, are there any molecular biology questions or current topics that you would like to see maybe answered in another decade or so?
I can think of one or two every second, but that's how my brain works. But so in 2010, we announced the first synthetic microbe. So we synthesized 1 million letters of genetic code. We actually made the DNA and booted it up to create a cell. So that's the first time, or the second time maybe in history that's happened. One of the big questions is that people have is on the real origin of life. So right now we used an existing cell with the DNA removed that we put the synthetic DNA into to boot up the chromosome. It made all new proteins, made a whole new cell, but the question is, could we make that from basic components if all the proteins could be isolated and purified? Things get back to what the original origin of life was. You know, was it from RNA, simple systems getting more complicated? Was it from high temperature vents? One of the things all the microbes in the oceans do is they do more complex chemistry than our best chemists can. And so one way to do that is because microbes use chemical warfare, to kill off their enemies and things. If we can take all the gene pathways from these millions of microbes, we could make whole new sets of chemicals, whether it's for medicines or construction. I have a few other ideas if you have time. So it's just ask simple questions about life. Where did it come from? Where did it go? How different are you from your parents? Yes, sir.
Can you just describe shortly how artificial intelligence and computational biologists affect genomics today compared to when you were with genomics and let's say the 90s?
The question was, how does artificial intelligence and computational abilities affect what we can do in science today? Well, my success in my career was by being the first to apply high-throughput computing to biology. And one of the reasons I started my own not-for-profit institute, I was very happy as a researcher in the intramural program at NIH. I had a multi-million-dollar budget each year based on a 30-minute conversation to get that budget. But I tried to get a top computer scientist promoted in my lab because I knew the computational approaches were the future of understanding biology. And the higher-ups at NIH said he's not a biologist. He should maybe go over to the computer core. No, you can't promote him. So I realized that the government was well behind on seeing what the future was. So I left and started my own institute that computing has been a big part of. Now, AI is, and if you've used ChatGPT, for example, all of AI is based on having a training set. ChatGPT uses everything on the internet, which is why it comes up with fake news 40% of the time. It's not a way to predict unknowns, because unknowns are not part of the repertoire it was trained with. So seeing that so much of biology is still unknown, it's been helpful. It's improved predicting three-dimensional structure of proteins, which people have been working on algorithms for a long time, but not as completely as people had hoped it would. So I think it's overrated for probably 99% of things people think it's going to be good for. It's not a substitute for the human mind making new associations and breakthrough ideas. It can be a tool that can help. Yes, sir.
I think you said earlier that your 6 billion letters in our DNA were all different. Than my understanding, I hope it's true, is that large percents of our DNA is identical to other species in very small differences, which account for us being different from a chimp or a protozoa.
Yes.
What is the significance?
Well, it's called evolution. We're related very closely to all the mammals. We have 2 dogs, a poodle and a golden doodle, who think they're more human, and quite often we prefer their company to some humans that we know. But we as humans differ from each other maybe 3 to 5% of our genetic code. all mammals differ by each other by at the most 10%. So the steps in evolution that led to us led to all the other mammalian species, and why we recognize emotions, intelligence, personalities in animals, because it's all part of where we came from in that journey. My genome, being the very first one sequenced, was used with some mirth by my colleagues, proving that the human genome still had Neanderthal DNA. So it was proven that a certain percentage of my genome was still had semblance of Neanderthals. Some people I know have a whole lot more, but groups of people that are from Africa and never migrated out and back had no Neanderthal DNA in their genome. So these things can be used to track history of populations. We're closely related genetically to every other species, including the microbes on this planet. But the diversity explains everything. So the computer code is ones and zeros, and we see everything that can be done computationally. Our genetic code is a 4-base system with A, C, G, and T, and subtle changes in those combinations make you different from your parents. My genome was the first diploid genome that was sequenced. My 2 parental chromosomes differ by 0.5% from each other. So we're all unique combinations, even identical twins. I don't know if anybody here is an identical twin. You're really not identical, and we can distinguish you from your twin by DNA sequence, because every one of us has a few million random mutations that make us totally unique and identifiable. So it's your history, it's my history, it's the history of all life on this planet. Other than that, there's no significance. All the way in the back.
Yep.
As I understand it, you've taken all of the results of your research and made them publicly available free of charge. Is that correct?
That's correct.
Then my question, so you've lost control of it. My question is, what's the best thing that you think has come of it, and what's the worst thing that's come of it?
Well, probably the best thing is just the broad idea of understanding that diversity is orders of magnitude greater than anybody imagined on this planet that we're trying to understand. And it's Heather's favorite term. When I use it, I talk about visual acuity. Darwin could not see microbes, so he didn't know they existed. You look outside, you can't see them. You look at the ocean. Some places in the South Pacific, the water is so crystal clear that easily several hundred feet visibility when you're diving. But each milliliter of seawater has 1 million bacteria and 10 million viruses in it. So, just understanding that we're sort of visitors in this microbe planet, and understanding that diversity is probably the greatest thing to come out of it, and at least intellectually to me, and that it was from simply asking a very basic question about life and diversity and sequence. The worst thing is that people continue to develop very naive ideas about human genetics and thinking, and then they go off to prove that they might be right sometimes. So, other than that, nothing negative has come out of it, to my knowledge. Yes, sir.
I'm just curious, when I have partly answered this question because you said that you tell whether something's a virus or not, but when you sequence a genome, I have to tell you, please, that this is a virus, this might be a bacteria, this might be a single cell organism, but how much more of a story does the genome tell you? Does it tell you what kind of proteins it creates or anything about how it might live its life?
Yeah. No, it's a very important question. So when we sequenced the very first genome in 1995, I was not happy or satisfied with the accomplishment of sequencing the first genome. And so we spent months before we published it trying to understand what the genetic code was telling us about the life of that organism. And yes, you can predict all the different proteins from the genetic code, but probably our biggest discovery at the time was about 40% of the sequences predicted for things that were unknown that hadn't been seen before. And this is the case with the human genome, it's with every genome we've done. So and this came to a head. I made the decision to try and make a synthetic life form to try and understand the genetic code. As Richard Feynman said, if you have to build it to understand it. And we actually had a contest at the Institute of people designing what they thought was the minimal genome, which proteins were necessary for life based on the entire scientific literature. And we made 2 or 3 different versions based on these predictions. They all failed. Nothing led to a life form. And we spent the next 5 years randomly adding back unknown genes to our synthetic genome until we could get a living cell. And it turns out for the minimal genome, the smallest number of genes that leads to any species that we know of, that we created, about 20% of its genes essential for life are of unknown function. And science keeps going through these bottlenecks where we think we know everything, and people stop diversifying their thinking. NIH doesn't like funding exploratory science like we did on this expedition. You have to be trying to answer a very specific question. As a result, science has probably missed 30 to 40% of all of biology that's essential for life. So we're still trying to answer that question. There's over 100 labs working on our minimal cell is the best tool in genetics, because the idea is we should be able to recapitulate all of evolution by adding functions back to this minimal cell. People thought they understood basic cell division. So the minimal cell, Jack Sozak, who got the Nobel Prize for discovering telomeres, worked on origins of life, building basically lipid soap bubbles. And we've all seen how bubbles sort of bleb off, and that's how this minimal cell originally divided. So, but all other cells on the planet have a very specific pattern of cell division. So we added back the known cell division genes, and they did not restore function. We had to add back, by trial and error, 3 unknown genes to understand cell division. We still don't know what these genes do. We just know you can't get cell division without them. So those questions are being asked and answered every day with the new tools that we develop. But, I started sort of where your question did, of thinking we should be able to interpret all of life by looking at the genetic code. I'm mainly focused on trying to do that with the human genome right now, and it's stunning to me in 25 years since I sequenced the human genome, it seems like almost we understand less than we did before. So, but thank you, it was a great question. Yes, sir.
So much of biology depends on the right thing happening at the right time in the right place. What are we learning about control of transcription?
Well, it's something people have been studying for, multiple decades, but it's interesting. So I was born in 1946. I know some of you were born before, but the entire field of starting with understanding that DNA was the genetic material started in 1944. And so basically everything that's happened with changing our understanding of the biological world, of genetics, has happened in my lifetime, or definitely in some of your lifetimes. And so this is all very recent history. I mean, it's like the tiniest blip in history, but understanding transcription is very key, and it's key to actually being able to boot up a synthetic chromosome. So we're trying to see if we, there's a lot of variations in genetic code. For example, mycoplasmas use a slightly different genetic code than the bacteria E. coli uses. So they have to have very specific different ribosomes, and the whole process of making proteins is subtly different. So it's still at early stages of trying to truly understand it, even though you can find biochemistry textbooks that I used in the early 70s that tell you we knew all about it. So stay tuned for another century or more. Yes, sir.
It's popularly in the scientific community, I've read, assumed that the water on the Earth came from asteroids. So if we could see water on an asteroid, it wouldn't have the microbes that you're discovering that are in the water on the Earth. These microbes are a product of the generation of life on the Earth.
Is that a question or an assumption? Okay, good. So I wrote about this in Life at the Speed of Light. So Earth and Mars exchange about 100 kilograms of material a year. So you can't go out here and take a shovel of soil without having some Martian soil in it. It's clear that there's water on Mars. It's clear that there were oceans on Mars. I'm certain there had to be life at least at one time there, because from things hitting the Earth and splashing and landing on Mars with the exchange, we would have impregnated Mars with microbes. Likewise, the idea of panspermia is that life originated somewhere else and came here and on an asteroid or some fragment, and things work here. We still don't know. I'm certain, as one can be scientifically, that wherever we have the right chemical milieu, that life will just happen, and that we will discover life probably in the subsurface oceans on Mars. Be more stunning if there wasn't life there. I think that would be a bigger discovery than discovering life. And if we discover life there, I'm sure it will match something in our DNA databases. That's a long way of avoiding answering your question. Yes, sir.
You've been talking all in the physical of the microbes, and so do you wrestle with the non-physical aspects of microbes?
what does that include?
Well, I mean, we as humans have feelings, and that is our non-physical realm. And I wonder if
Well, you assume that they are. Your feelings are actually chemical-based and genetically based. And yes, microbes can sense their environment. Nobel Prize went to a couple of people for something called quorum sensing, where they sense communities and respond to them. Life is chemical and everything is driven by the genetic code. So just because it's a thought in your brain doesn't mean it didn't derive from these basic fundamental pathways. Yes, ma'am.
Are there any ethical things that bother you about this information being out there, creating cells, trying to create cells?
It's a huge problem right now, and I'm on an ethical review board. I'm on a government panel that makes the rules for what can be exported out of the country. DNA sequence is a huge issue right now because China has a one-way path. Data can go in, but it can't come out of China. China has funded a company called BGI, Beijing Genome Institute, that has set up very cheap sequencing outside of major universities in the US and take all the data, and it's on a one-way trip to China. So there's all kinds of rules and laws being considered about how to either force an open exchange. I've had meetings with several senior scientists and political officials in China, and they all, to my face, say they have no problems with sequences leaving, but then they never do. So it's a real problem because many people are starting to believe these technologies are the future of our economy. Understanding the genetic code, I contend, is the future of medicine. I could go on for hours talking about what we're doing I set up a clinic to do pre-symptomatic testing of people, measuring a large number of things along with the genetic code, and only screening people that think they're healthy. And we find at least 50% of them have something substantially wrong with them. 5% of everybody over 50 has a major tumor that they're unaware of. 1% of the population has a brain aneurysm. But older people here, you're safe, because if you had one, you would have already died from it by now. So it's the population is skewed to people under 60 for that 1% of the population. These things are predictable, they're actionable, there are ways to change the medical outcome, and helping us to interpret the genetic code so these things will be predictable in the future. So, yes, ma'am.
Can you explain what caused the absence of oxygen in the water where you detected it? And what are the implications for the towers toxin that sounds in that?
I'm not an oceanographer, so I don't know how they originally originated, other than it was, increased toxic blooms of anaerobic organisms, quite often from fertilizer runoff or other things that caused huge phosphate concentrations going up, and there are blooms of different microbes that sort of took over and killed off the anaerobes. So, but there's probably 1,000 different reasons why they develop, because these environments are very complex. in the back, all the way.
Circling back to your comments about possible life on Mars, and it's interesting you mentioned that some of the viruses you find in the ocean are completely unrecognizable. Considering that, there's stuff coming in from space in various ways, how would you recognize an alien organism if you found one?
Well, I lived in Washington, D.C. for about 20 years. So there's some really amazing aliens there, but we wouldn't necessarily just on the sequence alone, because so many of the sequences, as you point out, don't match anything else. We sequenced all the viruses in human blood and found thousands of circulating viral sequences in human blood. That was another major surprise. So the only way you would do it is is there anything on our existing databases that they match, even if we don't know what they are? Do they clearly have the same genetic code? Do they code for the same amino acids? People mentioned asteroids earlier. We find all the basic chemicals of life, including the basic components of proteins and DNA, on asteroids. So to me, it's not a question of if we'll find things, it's more what we'll find. I'm hoping there's intelligent life out there somewhere. Do you want me to stop with that one?
Absolutely. Nah. No, excellent. That's the fine. Yeah.
Something good.
Like that. Something animals.
Before you stir, good friends, I think we set some records at this meeting, perhaps for attendance, looks like it to me. And I thought that was an amazing presentation on Craig Venner's part. I also have to say I was very proud of the questions that you asked. So there are some remarkable minds on this island, and every day, just like your microbes, we find a new one. So thank you for coming out. And let me just say a quick word about next week. We're going to stick with the seas in a certain way. We'll have an expert on naval intelligence and the strategies that these naval intelligence experts are recommending we follow in the South China Sea to counteract the actions that the Chinese are taking to assert their dominance in that area. So it should prove to be very interesting indeed. And we would welcome you as two new members of the audience. Hope you can make it. Thank you all for coming. And another round of applause for Craig Venter, please.